Hello students. So in last class I explained about outbreeding devices or contrivances for cross-pollination. So you know that cross-pollination is nothing but the transfer of pollen grains from anther to receptive stigma. So the plants have developed various mechanisms to achieve cross-pollination. So they are collectively called as outbreeding devices because cross-pollination is more beneficial for plants when compared to self-pollination. It is having some advantages. What are those advantages? So cross-pollination is that brings about or that results in variation because of mixing of genes in plants or the genes from different sources. They are coming together. So that's why variations were observed in the offsprings. So these variations which are necessary for the plants or any organism to survive in the nature. So variations, you know that variations that is leads to evolution or that is one of the cause for evolution. Okay. So to survive in the nature, to increase the survival ability of an organism, each and every organism that needs variation. Similarly, the plants also have adapted cross-pollination to achieve variation. So that is one of the purpose. Okay. And also it is having some disadvantages also because the plants has to develop some other structures or modifications to avoid self-pollination. Okay. So to achieve cross-pollination, the plants have developed outbreeding devices. So outbreeding devices like dicliny, dichogamy, hercogamy, heterostyly, self-incompatibility and self-sterility. So that I have explained in the last video. Okay. So in this class, I am going to explain about one of the interesting and also this is very important concept, that is pollen pistil interaction. Okay, so that is interaction between the male entity, the reproductive entity and the female reproductive entity. So how these interactions will take place in the plants, especially how a pollen grain that will interact with a pistil or how a pistil that can interact with a pollen grain. So that how it will result in fertilization or how it will result in the development of the seed or within that the embryo also. Okay. So that is what is called as, today I am going to explain about pollen pistil interactions. So not all pollinations that brings about compatible pollen grains on the stigma. Means the pollination does not result in the transfer of right type of pollen grains on the stigma. Whatever the way of pollination it may be, yave way pollination agidrunu ella pollinations anu right type of pollen grains na tandu stigma mele large madidaru. It may be biotic pollination also or it may be abiotic pollination also. So not all pollinations that carry the right pollen grains, or not all pollination even it is not carrying wrong pollen grains also. There are compatible or the right pollen grains are also transferred on the stigma because that is the main aim of pollination. But sometimes or usually there are some uncompatible pollen grains. It means the pollen grain of different species may lodged on the stigma. Okay. So means all pollinations may not be successful also. Okay. When pollination takes place, so along with the right type of pollen grain, the wrong types of pollen grains are also carried. The right type means the pollen grain that belongs to the same species, or even it may belongs to the same plant also. Okay, in case of the geitonogamy or in case of self-pollination. So that pollen grain it may belongs to the same species of different plants which are located somewhere else. Okay, it may belongs to the same species, that is considered as a right pollen grain. If it belongs the pollen grain, if it belongs to the same species. Sometimes if the plants wants to achieve cross-pollination, if pollen grain of the same plant that is considered as incompatible, that is what we call as self-incompatibility that I explained in the last class. Okay, so where though the pollen grains are functional, the stigma or the pistil that doesn't allow the pollen grain to germinate. That is what is called as self-incompatibility. So the pollination it may result in either falling of the compatible pollen grain, along with that the incompatible pollen grains also or the wrong type of pollen grains also which are fallen on the stigma. So next the question arises, does the stigma allow all these pollen grains to germinate? ಹಾಗಾದರೆ ಪಾಲಿನೇಷನ್ ಆಗಿ ಬಂದಿರೋಂಥ ಎಲ್ಲ pollen grains ಸ್ಟಿಗ್ಮಾ ಮೇಲೆ ಜರ್ಮಿನೇಷನ್ ಆಗುತ್ತಾ, ಎಲ್ಲದೂ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗುತ್ತಾ? ನೋ. the pistil that has developed some mechanisms that is able to recognize the right type of pollen grains. ಅಂದರೆ ಯಾವುದು ಪರ್ಟಿಕ್ಯುಲರ್ ಪೋಲನ್ ಗ್ರೇನ್ ಇದೆ, ಪರ್ಟಿಕ್ಯುಲರ್ ಸ್ಪೀಶೀಸ್ ಯಾವ ಪೋಲನ್ ಗ್ರೇನ್ ಇದೆ, ಆ ಪರ್ಟಿಕ್ಯುಲರ್ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಅನ್ನ ಪಿಸ್ಟಿಲ್ ಏನು ಮಾಡುತ್ತೆ, ರೆಕಗ್ನೈಸ್ ಮಾಡುತ್ತೆ. ದೇರ್ ಆಫ್ಟರ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗುತ್ತೆ. ಓಕೆ, ಸೊ there will be an interaction between the pollen grain and the stigma as well as the pistil. So when pollen grains that is fallen on the stigma, not all the pollen grains which are fallen on the stigma they germinates. Those pollen grains which are compatible or which may belongs to the same species, they will only allow to germinate on the stigma. And the other pollen grains which are mismatched or which are of wrong type, so they are not germinated on the stigma, or the pistil doesn't allow such pollen grains to germinate on the stigma. See, here we can observe two mechanisms. The first thing is recognition. The pistil is able to recognize whether the pollen fallen on the stigma, whether it is of right type or wrong type. So that is one thing. That is what is called as recognition. And another thing, if the pollen grain is of wrong type, such pollen grains are rejected. It means they were not allowed to germinate. And the third thing is the acceptance. If the pollen grain is of the same species, then the pistil allow the pollen grain to germinate. So these are the three abilities that the pistil is having. So first thing is recognition. The pistil recognizes whether the pollen grain fallen on the stigma, it is of right type or wrong type. Right type means pollen belongs to the same species. Wrong type means the pollen that may belongs to some other different species. Say for example, if a stigma of a hibiscus, if it is pollinated with the pollen grains of cotton plant, so no doubt hibiscus as well as cotton, they belongs to the same family, Malvaceae, but they are of different genus. Hibiscus is a genus is different, and even the cotton genus is different. So in such cases, the stigma of hibiscus that doesn't allow the pollen grains of cotton to germinate on its stigma. So the first thing recognizes. It recognizes whether the fallen pollen grain, whether it is of right type or wrong type. I think you came to know what is right and wrong. So right means the pollen grain of the same species is right type, and the wrong means the pollen grain of different species is wrong type. Sometimes in case of self-incompatibility, the pollen grains of the same flower, that is also considered as wrong type. It means the stigma doesn't allow them to germinate. So that is in case of self-incompatibility. Okay. So first thing it recognizes. Thereafter, if the wrong pollen grains, it means the pollen grain of different species, or sometimes the pollen grain of the same flower, it may be rejected. That is the second thing. And the third one is acceptance. Acceptance means the right type of pollen grains were allowed to germinate on the stigma. So such pollen grains, they germinates and produces a pollen tube, and that pollen tube will develops in the stigma and reaches a style and enters into the ovule. So where it releases a male gametes. So that is the accepted pollen grains. So the three mechanisms, the recognition, rejection and acceptance. Okay, so these three important things that taken place during the interaction between the pollen grain as well as the pistil. Okay, so how these interactions will take place with the pollen grain as well as with the pistil? These interactions that mainly or purely depends on certain chemical things or substances which are found in the wall of the pollen grain. So if the pollen grain of the same species, they have some chemical substances which are compatible to the pistil of the same species. It means there are some substances which are found in the pollen grain, the chemical substances found in the pollen grain. So they are compatible with, they interact with the substances found in the stigma. So it is purely the chemical interactions between the pistil as well as the pollen grain. So such pollen grains only which are having that compatibility, so they were allowed to germinate on the stigma. So if it belongs to same species, no doubt the chemical substances or the molecules or the compounds found in that pollen grain as well as in the pistil, they are compatible. If it is belongs to different species, so there will be an incompatibility among those chemical substances found in the pistil as well as found in the pollen grain. Okay, so that compatibility makes the pollen grain to germinate. So now what does this include? The pollen pistil interaction. What does this include? It includes just after the pollination, from the beginning of the pollination up to the entry of pollen tube into ovule and the release of male gametes inside the ovule. So once the pollen tube enters into the ovule, so where it releases the male gametes. So from the beginning of the pollination or just after the completion of the pollination process, from where up to the entry of pollen tube into ovule. So all these mechanisms together what is called as pollen pistil interactions. Sometimes if the pollen grain of the wrong type, if it may germinates on the stigma, even the pollen tube also produced, but the growth of the pollen tube in such cases is inhibited in the stylar region of the pistil. I am repeating, if pollen grain of the wrong type, sometimes even the wrong type of pollen grains are also germinates on the stigma, even it produces pollen tube on the stigma, but while entering the pollen tube into the stylar region, so the entry of pollen tube is inhibited in the stylar region also, and even sometimes in the regions of the ovary also. Okay, so where the growth and further growth of the pollen tube is rejected. So the main thing, the first rejection reaction that occurs in the stigmatic tissue and the second rejection reaction that occurs in the stylar tissue. Sometimes if they germinate on the stigma, their growth is inhibited in the stylar region. But when it is compatible, the pollen grains will germinate on the stigma. So where it produces a pollen tube, the pollen tube enters through the style, then it reaches the ovary. So within ovary, it finds its way towards the ovules. So where the pollen tube that grows and finds its way towards the ovule. Until here, all these reactions or mechanisms together what is called as pollen pistil interactions. Okay. See, then what is the role of stigma? The role of stigma here, it is very, very important. Because the stigma, it may exude some compounds. There are some chemical substances which are exudated or which are secreted by the stigma, and that mainly consists of the lipid compounds. The chemical exudates are the stigmatic secretions that mainly consist of lipids and phenolic compounds. Lipids and phenolic compounds. So these are the major contents of the stigmatic exudates. Along with this lipids and phenolic compounds, the stigmatic exudate, it may also consist of little amount of sugars. It also consists of some amino acids and proteins. So these are some of the substances which are found in the stigmatic exudates. So this mainly helps for the germination of pollen grain on the stigma. So now the question arises, what is pollen germination? So pollen germination is nothing but it's a process of formation of pollen tube through the germ pole. So you have studied the structure of pollen grain. A pollen grain that consists of two walls. The outer wall is what is called as exine and the inner wall is known as intine. So during the germination process, the entire content of pollen grain that is comes out in the form of a tube. It come out in the form of a tube, okay, through the germ pore, and that mechanism is what is called as pollen germination. So during pollen germination, the first prerequisite for the germination is hydration of the pollen grain is very much necessary. So it means the pollen grain absorb water that is found in the stigmatic exudate or that is found in the stigmatic surface. So once it absorbs the water, the outer wall of the intine that absorbs water and that swells up. And it swells up and applies the pressure on the wall of the exine. So whereby the wall of the exine that ruptures and the entire content of the pollen grain will come out in the form of a tube. That is what is called as pollen germination. Pollen grain absorb water, that is what is called hydration. It absorbs water, the wall of the intine that swells up, it applies pressure on the exine. So where at that time the entire content of the pollen grain that come out in the form of a tube, that is what is called as pollen germination. Okay. So for pollen germination, all these substances which are required, they play a very major role in germination of the pollen grain. And also these secretions, they consist of some of the elements like calcium and boron. There are some elements which can observe in these secretions, inorganic elements like calcium and boron. So they also play a vital role in growth of the pollen tube and in directing the pollen tube in the pistil. Okay. So how a pollen tube that moves in the pistil, that is guided by or directed by calcium ions. So boron, that helps in vigorous growth of the pollen tube and also prevents the pollen tube from bursting. Okay. So these two elements also very important in growth of the pollen, in germination as well as in the growth of the pollen tube. Calcium ions, they direct and helps in elongation of the pollen tube. They direct the pollen tubes in the pistil, as well as boron helps in vigorous growth, a rapid growth of the pollen tube, and it prevents bursting of pollen tube. So whereby the pollen tube can enter into the stigma, style as well as finally it enters into the ovary. Then it enters into ovule. So that is the role of stigmatic exudates in pollen germination. So that is one thing. So it means pollen pistil interaction starts just after the pollination. So begins with the germination of the pollen grain. So then pollen tube enters through the stylar canal, it grows through the stigma, enters through the style, then it reaches the ovary, then into ovule. So all these mechanisms here, they were collectively what is called as pollen pistil interactions. So I said that pollen pistil interaction is mainly due to the chemical substances which are found in the wall of the pollen grain or in the pollen grain as well as in the stigma. They recognizes if it is whether it is compatible or if it is incompatible. The compatible pollen grains only they germinate on the stigma. The incompatible pollen grains, they were rejected. So those pollen grains are not allowed to germinate. If it is germinated on the stigma, if the pollen tube enters the style, then the rejection reaction will occur in the stylar region. If the compatible pollen grain that germinates and it transfers through the style, the pollen tube transfers through the style. So where the pollen tube that carry the male gametes and that male gametes will be releases inside the ovule. So this is one of the most interesting mechanism that takes place in the plants, especially in the agiospermic plants. So and the knowledge of this, the pollen pistil interactions, the knowledge of this chemical reaction or the interaction is very much important. How it may be very much helpful in breeding, plant breeding works. Okay, sometimes the self-incompatibility that can be overcome by knowing or by having the knowledge of this one, pollen pistil interaction. So we are making or we are forcing the self pollen grain to germinate on the stigma. In case of self-incompatibility, it can be overcome. The self-incompatibility can be overcome. So by treating with some chemicals, so where the self-pollen grains can be made to germinate on the stigma. If you want to have self-pollinating plants, so in such cases we can make the pollen grain to germinate on the stigma. So then as usual the fertilization takes place. So that's why the knowledge of pollen pistil interaction is very much important. So the overall events that takes place between the beginning of or the just after the pollination up to the entry of pollen tube into ovule is together what is called as pollen pistil interaction. The role of stigma is very important here. Stigma that secretes some of these chemical substances together called as stigmatic exudates that mainly consists of lipids and phenolic compounds. So as well as the sugars, amino acids and proteins. So main thing, the hydration of the pollen grain is very much necessary in or during the germination. So thereafter the pollen grain, the intine that swells up and applies the pressure and the exit where pollen tube come out. So not only one pollen tube that come, usually only one pollen tube that come out or one pollen tube that grows from a pollen grain. That is usually in usual cases. In some of the cases, for example, in plants like a member of Malvaceae, Althea rosea, where we can observe 10 pollen tubes. We can observe 10 pollen tubes from a pollen grain. So in plant like Malva neglecta, so where we can observe 14 pollen tubes from a pollen grain. Usually one pollen tube that grows from a pollen grain, but in rare cases, in some of the species. So more than one pollen tube may grow from a pollen grain. So example like in Althea rosea, so 10 pollen tubes will come out from a pollen grain. So in Malva neglecta, 14 pollen tubes may come out from a pollen grain. Okay. So these events, they were together called as pollen pistil interaction, and even it will be asked in the examination for 5 marks, write a note on pollen pistil interaction. So this is purely the chemical interaction. It's a very long process. So I already said that the 3 important mechanisms. So one is the recognition, the rejection and the acceptance. The pistil is able to recognize whether the pollen grain is compatible or not. And the second thing, it rejects if the pollen grains are incompatible. And the third thing, it accepts when the pollen grain is compatible, and such pollen grains they germinate, thereby it leads to fertilization. Okay, so this is about pollen pistil interactions. The next concept is artificial hybridization. See, hybridization is a process where crossing between the two organisms having contrasting characters. So when it comes to plants, hybridization is a process of crossing between two plants having contrasting characters. It means the two plants are differing in some of the characters. Whether it may be one of the plant may be tall when it refers to height. So one it may be dwarf. Okay, so one of the plant that may bears very larger fruits, another may bear a very smaller fruit. So these are the two contrasting characters when it comes to the size of the fruit or when it comes to the height of the plant body, whatever it may be the character. So when two plants are having contrasting characters, so we are crossing them. Okay, that is based on the conventional or the traditional plant breeding methods. It means we transfer the pollen grain from one plant to another plant. That's why it is said to be artificial hybridization. We select the pollen grains from one plant, and those pollen grains will be transferred or dusted on the stigma of the another plant. Why? That is to get the hybrids or to produce the desired characters. What are the characters which are required for us? The main thing is, the purpose of artificial hybridization is to increase the yield. Any plant breeding purpose, it may be conducted to increase the yield. That is one of the major aspects. The yield has to be increased, because nowadays the demand will be more. For increasing population, the world has to supply the food for each and everyone. For that, the agricultural fields also, they should be develop different mechanisms to get more yield. So, and another thing, one is for the yield, another thing to make the plants disease resistance, okay, or to make the plants resistant to some environmental conditions. It may be drought or it may be some kinds of stress. So for that we are following different mechanisms in hybridization. So that is one of the major aspects of artificial hybridization or any kind of hybridization. So nowadays the major problem is providing food for everyone. For that the yield has to be increased. So to increase the yield, so where we are following different methods of hybridization, we are doing conventional plant breeding methods. We also adapted the biotechnological methods like tissue culture or micropropagation to get or to produce many number of plants. Okay, so what is hybridization? I explained. So it is a process where crossing between two plants are having contrasting characters. Okay, the characters should be different in plants. So those characters are combining together in a hybrid. So where that hybrid that may bear the characters of both the parents, or it may bear the superior character. It means among the two parents, which parent is having superior character. So I think you have studied in your previous classes like the experiments of Mendel's. So like he crosses tall and dwarf plants. So where the tall is produced in the F1 generation in the hybrid. So that is a superior character. Tall is superior over the dwarf. So where combination of the two character, the tall as well as the dwarf, and they are coming together in a hybrid. So how Mendel he conducted, where he followed the same mechanism, what is called as artificial hybridization. He selected pollen grains from one plant, so where those pollen grains will be dusted on the stigma of the another plant. It means of the same species. Okay, so where that is what is called as artificial hybridization. See, this artificial hybridization that mainly includes two principles. So one thing is emasculation and the another one is bagging. So the first thing is emasculation and the second thing is bagging. So what is emasculation? Emasculation is nothing but it's a process of removal of anther from a flower bud. The process of removal of anther from a flower bud to avoid self-pollination. To avoid self-pollination. So why emasculation is conducted? See, if a flower is bisexual, in such cases, the anthers or the stamens has to be removed to avoid self-pollination. When we select bisexual species, emasculation is required. So, when emasculation should be conducted? Emasculation is conducted in the bud stage itself, in the flower bud itself. With the help of forcer, we are removing the anthers carefully without damaging any other whorls of the flower, especially the pistil. So, we carefully remove the anthers from a flower bud in a bisexual flower to avoid self-pollination is called as emasculation. Emasculation is followed in bisexual flower. No need of emasculation in unisexual flower. When male and female flowers are separate, then no need of emasculation. Okay. So then emasculation should be followed by bagging. We cover the emasculated flower with a bag made up of a butter paper. So we will cover the flower with a bag. So why the bagging should be done? So it is to avoid pollination from unwanted pollen grains. If there may be some other pollen grains which may fallen on the stigma, so that is what we have not selected. So that pollen grain may fallen on the stigma and that may pollinate or that may fertilize the ovules. So in order to avoid this pollination from unwanted pollen grains, the pistil or the flower that should be covered with a bag, usually made up of butter paper. So that is what is called as bagging. These two are the principles, emasculation as well as bagging are the principles of artificial hybridization. See here, we are selecting two plants. If they are bisexual, we are selecting two plants. Among that, one is considered as female and another one is considered as male. So the plant which is considered as female, from that the stamen should be removed. We are selecting two plants. They are bisexual. Here, no doubt for hybridization work, two plants are required. If male and female plants are separate, or male and female flowers are separate, no need of emasculation. If we select a bisexual species, among that one is considered as female, from the female plant, from that flowers, the anthers should be removed. Emasculation should be done. And where from the male, the plant considered as male, so from that the pollen grains are collected and they were dusted on the stigma and again they were rebagged. Okay, so where after the pollination, we dusting the pollen grains on the stigma of the emasculated flower, then we rebag. After the pollination, there will be a fertilization and there will be a seed setting was observed. So that is the principle of artificial hybridization. So it includes two mechanisms or principles. One is emasculation, another one is bagging. Okay. Okay, so in next video, I explain about the process of double fertilization. Okay, so one of the most important mechanism that is found only in flowering plants or found only in angiosperm. So up to here you go through the pollen pistil interaction as well as artificial hybridization. In my next class I explain about double fertilization. Okay, thank you.